All right, ladies and gentlemen, welcome back. We're going to pick up our discussion of protein synthesis by talking through the process of translation today. So remember, translation is using that mRNA transcript that we just produced during transcription and converting it into a polypeptide, into a protein chain. So we've gone through that process in the nucleus of transcription where I've built that mRNA molecule. I've processed it, I've edited it. If we're talking about a eukaryotic cell, I've left the nucleus, and now I'm going out to the ribosomes located in my cytoplasm. If this is a protein that your cells are going to produce for itself, then more than likely it's going to go ahead and produce that protein on a ribosome that's free floating in the cytoplasm. If this is a protein that needs to get packaged in some way, whether it's because it's for something like digestive enzymes in a lysosome, or it's a protein that will ultimately get excreted out of the cell, then we will probably build that protein on a ribosome that is attached to your RefER. The other cool thing here is that in order to be a bit more efficient, your cells can actually attach multiple ribosomes along a single mRNA molecule, like you see here in the picture. It just speeds up the process, allows us to use less resources if I can use a single copy of mRNA to build multiple protein chains, and if I can do it all kind of simultaneously, or more or less simultaneously, then just speeds up the efficiency of the process. So before we jump into the actual steps of translation, let's take a step back and remind ourselves of the key molecules here that I need in order to make translation successful. So first and foremost, you're going to need that messenger RNA, because that messenger RNA molecule is what holds that copy of the code that you created in transcription from that DNA molecule itself. Also remember that we are reading this messenger RNA in triplet nucleotides. It's a triplet code, what we call a codon. So every 3 nucleotides, every codon, then corresponds to a single amino acid. Another key player are your tRNAs, or your transfer RNAs. Remember, these guys are going out into your cytoplasm. They are searching for amino acids and attaching them, but they are not just randomly pulling amino acids. Instead, that amino acid that is binding up here at this site corresponds to the anticodon that's down here. And remember that that anticodon is then corresponding to the codon in your messenger RNA. So if I have a UGG anticodon, if I remember my base pairing rules for RNA, then U goes to A, so I have an A in that initial space here of my codon, and 2 G's will correspond to 2 C's. So I have an ACC as my codon that's complementary to that anticodon, and that anticodon codon are then complementary to the amino acid that's being bound up top. So because there are 20 different amino acids, that means there has to be at least 20 different tRNAs with 20 unique anticodons that correspond to 20 unique codons. There's more than that. There's actually redundancy in the code, which is good. We'll talk a little bit about that when we get to mutations, but that's sort of where we're at right now. So then you've got your ribosomal RNA, your rRNA, and these guys, along with proteins, are assembled in the nucleus, which is in the center, your nucleolus, which is in the center of your nucleus. And that ribosome is built there into 2 pieces. So it kind of reminds me of a burger roll or a burger bun, in the way that it's shaped, typically in diagrams, and the way that it's ultimately actually put together. So you've got these 2 subunits here. You've got this small subunit down here, and then your large subunit up here. And your small subunit kind of sandwiches your messenger RNA here, and then between the large subunit as well. Within that subunit, you've got these 3 binding sites. They actually get used in this direction, so A comes first. It is an attachment site where we're going to do some codon recognition. So I'll have my messenger RNA code down here, and we're looking to find that complementary tRNA codon up here so that when they match, they bind together. My P site here in the center is a peptide bonding formation site. So once that tRNA anticodon matches its codon and messenger RNA, we slide everything over into this P site, and that's where we start building those peptide bonds. Remember, peptide bonds are the covalent bonds that get formed between amino acids in a protein chain. And then you've got your final bonding site here, the E site, which really is kind of not a bonding site. In fact, it's the bond breaking between the anticodon and codon once that amino acid gets attached. So I end up with a tRNA here with no amino acid, and it gets bumped back out into the cytoplasm, goes and finds its corresponding amino acid again so that it can continue to do its job. So when we look at the actual process of translation, let's take it step by step. So first and foremost, I've got to start with this sort of pre-translation. What sort of things have to occur before the real translation process can happen? So first and foremost, I need to build that mRNA molecule in my nucleus. I have to go through transcription. That mRNA needs to be processed. It needs to leave the nucleus. It needs to migrate to that ribosome, either ribosome that's freely floating, depending on if I'm going to be using that protein somewhere in my cell, or it goes to your RePER and attaches to a ribosome that's there. If it is something that needs to be packaged into a vesicle for my cell to use, or if its ultimate destination is to be exported from that cell and be used somewhere else in the body. In addition to while all of that migration is happening, your tRNAs are hanging out in your cytoplasm, picking up their corresponding amino acids. Where are those amino acids coming from? Well, they're floating around your cytoplasm because somewhere along the way, your body has digested proteins, broken them down into those corresponding amino acids from things like the foods that you eat. Once those tRNAs are properly connected to their amino acids, they'll then move to the ribosomes in hopes of finding a matching codon. In addition to all of that, I actually have to build this RNA ribosome complex. So what does that end up entailing? Well, I have to sort of build the sandwich, if you will. So my small subunit here sandwiches in between the messenger RNA and this large ribosomal subunit. But I need to do this in a very strategic way. So when we look at this codon chart in just a few moments, you're going to see that there's a very specific 3-letter code that tells your ribosome this is where the start of the actual protein information begins. Because if you look, here's my messenger RNA, but I've got all of these nucleotides ahead of that actual start codon, which is AUG. So I need to line this up so that that AUG start codon is in line with that P site. Everything else before it won't become part of the information utilized to make the protein, but everything after it will. So once that is attached there and I have the corresponding tRNA match that occurs, which will always carry a methionine, by the way. All right, so AUG always codes for MET or methionine. That's all bound into that P site. Large subunit comes on, finishes the sandwich, and now I'm ready to go ahead and do the next step. And that next step, so here's the initiation that's part of that building of the sandwich. The next step is the codon recognition. And that codon recognition is always happening here. This is your A site. So that tRNA is bouncing in here in hopes of finding a match. So you'll have random tRNAs that bump in. If they don't match the codon here, based on the anticodon that they carry, then they get pushed back out and the A site is left blank. Another tRNA comes in. If it doesn't match, it gets bumped back out. Eventually, you'll get a tRNA that will bump in here, and it will have the corresponding match, which is what you're seeing here. Once that happens, then we go to step 4, which is the peptide bond formation. So the amino acid that is currently hanging off of here, that MET, now gets attached to the tRNA that's attached here in the A site, and we form that peptide bond form. When that occurs, everything is going to get shifted over, which is what we see here. So here's that tRNA that was just here in this A site a moment ago. It gets shifted over, and now this guy who had left its amino acid behind ends up in that E site and is blank. Because it no longer has that amino acid attached, the bonds that were going on here between codon and anticodon get weakened, and that tRNA gets kicked out, kind of like hitting an eject button. This tRNA, now that it's blank and has no amino acid attached, goes out into the cytoplasm, searches for its amino acid again, so that it can continue the process. And this then leaves the A site open with a new codon. So we're back to attachment. Can I find its matching code? Once the anticodon codon match, then this set of proteins comes over, builds that protein polypeptide chain, shift everything over. This guy now ends up in an E site because it's blank and gets kicked back out. And we'll continue to open up that A site for recognition, attach amino acids together, push out the blank tRNA, and that keeps happening during elongation until I've gotten through all of the codons in my mRNA. Eventually, we'll reach a stop codon, and when we look at that codon chart, there's actually 3 stop codon potentials. When that stop codon ends up in that A site, we actually bind what is called a release factor, and that release factor just blows this whole complex apart. So the large subunit falls off, my protein chain comes apart, my tRNA gets kicked out, small subunit falls apart and releases the mRNA, and now all of these pieces are off to do the next step.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid, the first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.